Hello friends, myself I am Praveena from PhD College of Arts and Science. I am currently doing my Masters MSc in Botany. So today I am here to discuss about the topic microbial growth and measurements as in the subject applied microbiology. So let's start. Microbial growth curve. We know the microorganisms grow and reproduce by binary fission. So the growth of microorganisms is represented in the microbial growth curve. It is a graph that showing the logarithm of the number of viable cells versus the time or incubation time. So this is the microbial growth curve in a closed system. It contains four phases or the microbial growth curve or the four patterns of bacterial growth are: first one lag phase, second one exponential phase or log phase, third one stationary phase, fourth one death phase. By looking into the graph, it is a graph that showing that number of viable cells in the log phase against the time. So the growth of the microorganisms start with the lag phase, that is the bacteria or microorganisms starts to grow and there is a increase in the phase that is called exponential phase or log phase, and next stationary phase, that is the cells remains constant, and the death phase that the microorganism starts to death. We can look into detail manner in each phases. The lag phase, when microorganisms are introduced into the fresh culture medium, usually no immediate increase in cell number occurs, so the period is called lag phase. Although cell division does not take place right away and there is no net increase in mass, the cell is synthesizing the new components. The lag phase varies considerably in length with the condition of the microorganism and the nature of the medium. This phase may be quite long if the inoculum is from an old culture or one that has been refrigerated. Inoculation of a culture into chemically different medium also results in longer lag phase. On the other hand, when a young, vigorously growing exponential phase culture is transferred to fresh medium of the same composition, the lag phase will be short or absent. Log phase. During the exponential or log phase, microorganisms are growing and dividing at a maximal rate possible given their genetic potential, the nature of the medium, and the conditions under which they are growing. Their rate of growth is constant during the exponential phase, that is, the microorganisms are dividing and doubling in number at regular intervals. Because each individual divides at a slightly different moment, the growth curve rises smoothly rather than in discrete jumps. The population is most uniform in terms of chemical and physiological properties during this phase. Therefore, exponential phase cultures are usually used in biochemical and physiological studies. In the stationary phase, the total number of viable microorganisms remains constant. This may result from a balance between cell division and cell death, or the population may simply cease to divide but remain metabolically active. Eventually, population growth ceases and the growth curve becomes horizontal because this is a closed system. Microbial populations enter the stationary phase for several reasons. It is due to the nutrient depletions or the toxic substances also. The decline in viable cells of following stationary cells was described simply as the death phase. The bacterial cells were even transferred to the fresh medium. No cellular growth was observed because loss of viability was often not accompanied by a loss in total cell number. It was assumed that cells died but did not lyse. These are all about the microbial growth curve and the patterns of microbial growth. So next we are going to look the measurements of microbial growth. It contains the measurement of cell numbers and measurement of cell mass. First one, measurement of cell numbers. It is done by direct counting, membrane filtration technique and plating methods such as spread plate or pour plate techniques. By measurement of cell numbers, the first one is direct counting method, and it is used in a counting chamber. So it is very easy and inexpensive and very quick. It can be done by direct counting. So it also gives the information about the size and morphology of microorganisms. Petrov-Hauser counting chambers can be used for counting the prokaryotes. Hemocytometers can be used for both prokaryotes and eukaryotes. We can see here the Petrov-Hauser counting chamber. This is the picture showing the Petrov-Hauser counting chamber. So the prokaryotes are more easily counted in these chambers if they are stained or when a phase contrast or a fluorescence microscope is employed. The number of microorganisms in a sample can be calculated by taking into account the chamber's volume and any sample dilutions required. One disadvantage to the technique is that the microbial population must be fairly large for accuracy because such a small volume is sampled. This is the counter method. The large microorganisms such as protists and yeast can be directly counted with electronic counters such as the counter, although more recently the flow cytometer is used. The microbial suspension is forced through a small hole or orifice in the counter. An electrical current flows through the hole and electrodes placed on both sides of the orifice measure its electrical resistance. Every time a microbial cell passes through the orifice, the electrical resistance increases and the cell is counted. The counter gives accurate results when with large cells and is extensively used in hospital laboratories to count red and white blood cells. It is not as useful in counting bacteria because of interference by small debris particles, the formation of filaments and other problems. Next one is membrane filter technique. The number of bacteria in the aquatic sample is frequently determined from direct counts after the bacteria have been trapped on special membrane filters. So it is used in the aquatic bacteria, and in this membrane filter technique the sample is first filtered through a black polycarbonate membrane filter. Then the bacteria are stained with the fluorescent dyes such as acridine, orange or DNA stained API and observed microscopically. The stained cells are easily observed against the black background of the membrane filter and can be counted when viewed with an epifluorescent microscope. Plating methods for measurement of cell numbers. Several plating methods can be used to determine the number of viable microbes in a sample. These are referred to as viable counting methods because they count only those cells that are alive and able to reproduce. Two commonly used procedures are the spread plate technique and the pour plate technique. In both of these methods, a diluted sample of bacteria or other microorganism is dispersed over a solid agar surface. Each microorganism or group of microorganisms develop into a distinct colony. The original number of viable microorganisms in the sample can be calculated from the number of colonies formed and the sample solution. And the next is the measurement of cell mass. It is done with the microbial dry weight and the spectrophotometry. So, coming to the measurement of cell mass, first method, dry weight. It is the technique that the cells growing in liquid medium are collected by centrifugation, and it is washed, dried and oven and weighed. This is an especially useful technique for measuring the growth of filamentous fungi. It is a time consuming, however, and not very sensitive because bacteria weigh so little. It may be necessary to centrifuge several hundred milliliters of culture to collect a sufficient quantity. And the next technique by the cell mass measurement is a spectrophotometry. These methods are more rapid and sensitive, and they depend on the fact that microbial cells scatter light and strikes them. Because microbial cells in the population are of roughly constant size, the amount of scattering is directly proportional to the biomass of cells present and indirectly related to the cell number. When the concentration of bacteria reaches about the 10 million cells per milliliter, the medium appears slightly cloudy or turbid. Further increase in the concentration results in great turbidity and less light is transmitted through the medium. The extent of light scattering can be measured by a spectrophotometer and is almost linearly related to the cell concentration at low absorbance levels. Thus, population growth can be easily measured as long as the population is high enough to give detectable turbidity. So, happy learning. Thank you.